about the chemical compounds that make up living organisms. So if your Chromebook is open, shut it. Everybody should be taking notes. Everybody should be writing stuff on paper. And what I'm going to walk you through over the course of the next several weeks is the process that you should use in this class for getting information from a slideshow to your notebook. I'm doing it all right now, telling you just what to write down. Then after a couple more weeks, the responsibility is going to shift more to you, but I'm still going to kind of lead the show. And then we're going to switch gears, and I'm going to only focus on a couple of things. So if I put together a presentation, the understanding is going to be that there's information from there that should be in your notes, and I'm going to go over the stuff that you can really benefit from my help with. So I will go through the stuff that's not clear, concepts, processes that need to be reinforced, but I'm not going to go through and define every single term because that's not necessary and it's not really all that useful for you. It's not a good use of your time. So today we're going to talk about macromoleculars and focus on carbohydrates. And we'll start by just talking about macromolecules in general. So these are, the name macro means big. You do not need to write that down. But in the context of biology, what big implies is that we have molecules made up of carbon chains. That's the only way to get big in biology. We don't have any other good way of hooking things together to make things big. So when we talk about big molecules, we're talking about carbon chains. And there's only a few main groups. Now, there's literally millions and millions and millions of different ways that these things can be arranged. But in general, they kind of make there's patterns that we see. And so our work over the next couple of weeks is going to be to investigate some of the basic parts of those patterns. And one of the patterns is that many of these molecules, if you look at them, you can see, oh, there's this little piece hooked to this little piece, hooked to this little piece, hooked to this little piece. These are all the same. And in chemistry, when you have small pieces that are hooked together to make a chain, the little pieces are called monomers, and the longer chain is called a polymer. So this is something you probably learned in chemistry. If not, I know I brought it up in my bio classes. You might have learned it in Mr. Barron's or Ms. Key's bio classes as well, if you took those here. The kind of standard analogy for a polymer is a train. And the monomers of a train are the train cars. You have one train car that's as small as it gets. You can't subdivide that any further. You can make as long of a train as you want by hooking together as many train cars as you want. And so we'll see that many of the molecules that we're looking at are molecules that can be polymers. We can create them from smaller repeating subunits. The four main categories, and we'll look at all of these, but we're just going to look at carbohydrates today. The four main categories are nucleic acid, DNA, and RNA, and those are polymers. So those are, we'll be able to identify discrete repeating subunits for those. The next group, and this is the one that we'll look at today, is carbohydrates. Those are made up of, those can be polymers. If they're not polymers, then they're just the basic subunit called a monosaccharide. And then as soon as you start hooking those monosaccharides together, you create a polymer. So carbohydrates can be, they can function as the single subunit, or they can be hooked together in repeating chains of that subunit. And then lipids, and when we get there, we'll talk more about this, but lipids aren't really polymers from a chemistry point of view. If you look at lipids, you'll see things that look like maybe they're sort of repeated, but they're not. And so if you look online, if you ask Google the question, are lipids polymers? Some people say yes. Most people would say no. Most people who really understand chemistry say no. I'm not a chemist, but I trust the chemists to tell me whether something's a polymer or not. So we're going to say lipids aren't a polymer. And then the last category is proteins, and proteins are made up of repeating subunits, so those are polymers. And you looked at all of these in bio, so none of this should be super mysterious. It's okay if you don't remember anything right now. We'll go through all of these one by one. The next thing that I'm going to show you is there's a ton of information here. You don't have to write any of this down. If this is useful to you, you can, at some point later on, you can save it to your Google Drive, or you could copy it into your notes, but you could also find this or something like it elsewhere. And basically what this does is just kind of break down a bunch of useful pieces of information to know about all of these different kinds of molecules. The main name, what we call the monomer, and what we call the polymer for those that exist as polymers. Yeah.
Wait, so like looking at that, does that mean that proteins, they don't usually just have like super long, just like carbon chains, but they're like the repeating ones that have some carbon chains?
Yeah, so they are carbon chains, but what you'll see is repeating. So proteins are made up of amino acids, and amino acids are carbon chains. And then to make a protein, those carbon chains get hooked to each other. So it's chains of carbon chains hooked together that can be thousands and thousands of amino acids long. And so it's got the names of the monomers and the polymers, and then it tells you the principal elements that are in each of these proteins. Also have sulfur, so I would include that there. And then a diagram or a molecular diagram or a drawing of the structure that talks about the functions and examples. This is useful, but mostly what I want you to get from this is a target. By the end of September, thereabouts, maybe a little bit after that, you all should kind of know this stuff. Not necessarily have it memorized, but know it. So like if we're talking about nucleotides, it shouldn't have to be something where you think, okay, what's a nucleotide again? Oh yeah, okay, that's a monomer, and that's part of nucleic acids. Okay, got it. It should just be, it's part of your vocabulary. And talking about monosaccharides and polysaccharides, and talking about amino acids and polypeptides and peptide bonds and the different elements that are in these things, it should be just part of your thought process. It's not something that you memorized. It's not something that you can recite on demand. It's something that if I ask you, hey, what kinds of elements will you find in DNA? You're going to know, well, DNA has nitrogenous bases, so there's nitrogen in there. DNA has a phosphate group, so there's phosphorus in there. And then everything has carbon, hydrogen, and oxygen. So I would expect to see behind me that nucleic acids have C, H, O, N, and P as their elements. And they do. I didn't have that memorized. I just know what nucleic acids are. That's the level of familiarity that you're going to need. You're not going to be doing recall. You're going to be speaking biology. You're learning a language. Okay. So this is, like I said, this is a target, not something that you need to memorize. Okay. And then let's talk specifically about carbohydrates for just a bit. So carbohydrates, the easier name that most of you have used are sugars, and those are interchangeable. Sugars isn't wrong, but we're going to use carbohydrates because it's the more official, formal, scientific name. These are used for a variety of purposes, and we'll talk about all of these in just a little bit more detail, but primarily they provide energy for organisms, they are structural, and they're used for signaling purposes. Signaling means communication. It's not communication from one organism to another, it's communication from a cell to another cell, or even some sort of chemical pathway within a cell. And let's see. So the monomer of sugars is called a monosaccharide, and it's just a simple carbon chain. It can have 3 or 4 or 5 or 6 carbons in it. There's 6 carbon sugars. One of the most important ones is a 6 carbon sugar. And then if you take those monosaccharides and hook them together, then you get the polymer, which is called the polysaccharides. Some of the most important carbohydrates only have 2 monomers hooked together, so they have a special name, disaccharide. Di means 2. But that's not something where, you don't have to worry about losing points because you don't know when to use poly and when to use di. Usually if there's more than one hooked together, you just call it a polysaccharide. Technically, a disaccharide isn't a polysaccharide because poly means many, di means two, and two isn't many. But that is really splitting hairs and is not all that important for us to think about. Let's look at these functions in just a tiny bit more detail. I mentioned that they can be used for energy storage, and like all energy storage in living organisms, it happens in the form of chemical bonds. So the way I think about it, this is not accurate from a chemical or physical standpoint. The way I think about it is that you have like a little spark, and when you have a molecule and you have a chemical bond, there's a little spark trapped in that bond. And if the bond gets broken, that little spark gets released. It might get captured again and caught up in another bond, or can be used for something else. There's a whole bunch of things that you can do with that little spark, but you can kind of picture it as being trapped in the chemical connections between elements and between molecules. And that is not accurate, but it's a useful way to think about it, and that's good enough for us. Okay. Another function of carbohydrates is structural. One example is that carbohydrates are a primary component in cell walls in organisms that have them. And we haven't talked about that yet, we will, but bacteria, fungi, plants all have cell walls. So there's lots of carbohydrate in their cells. That is for structural purposes. There's other structural carbohydrates as well. One of the main carbohydrates in plants is cellulose. And that's what makes up the tough part of plants. Structural components are often proteins or carbohydrates. And then the last thing, and this is related to the signaling that I talked about earlier. We get carbohydrates that are embedded into the membrane of cells, and then those basically serve as identifiers or ways for the cell to receive or send information to other cells in an organism or even in other organisms. Yeah.
Does like the OSE ending mean like the carb, like cellulose?
Yeah, cellulose, ribose, deoxyribose, glucose, fructose. Yes, OSE is the ending for sugars. Yep. And unfortunately, that is so. If you see OSE, pretty much it's going to be a carbohydrate. But if you don't see OSE, it might still be a carbohydrate. So like starch is a carbohydrate. Glycogen is a carbohydrate. So there are carbohydrates that don't end in that, but those can be hints. Here I'm going to go through some examples of carbohydrates because I want you to kind of have an idea of what some of them are and what their names are. None of this stuff is stuff you need to memorize now. All of this is stuff that you should have at least some familiarity with, and some of these things we'll dive into more depth. Here are two sugars. These are each 5 carbon sugars. They're really similar. You can see they look really similar. One is called deoxyribose and one is called ribose. Does anybody recognize these? You've heard these names before, yeah.
They're the D in DNA.
That's right. They're the D in DNA and the R in RNA. Yeah. Deoxyribose is the sugar that forms part of a nucleotide in DNA, and ribose is the sugar that forms part of a nucleotide in RNA. And the difference between these chemically is that ribose has an oxygen here. It has a hydroxyl group here attached to one of the carbons that deoxyribose doesn't, missing that oxygen. That's it. That's the only difference between ribose and deoxyribose. And that chemical difference leads to a difference in function such that DNA serves a completely different purpose from RNA. It's kind of crazy. And we'll talk a lot more about this. Shape plays a huge role in how biochemicals function. Shape determines what parts of a molecule are available for chemical reactions, and that determines what they do. So the shape and the function are really closely related. You don't need to memorize these now, but ultimately You should have a familiarity, like if I say deoxyribose, you should know it's a 5-carbon sugar, it's part of the backbone of DNA. If I say ribose, 5-carbon sugar, part of the backbone of RNA, you should know that deoxyribose is missing an oxygen somewhere. You don't need to have the structures memorized, but you should understand that these are carbohydrates, monosaccharides, that are also part of nucleic acids. Here's some other examples, and this is highlighting something that you learned in chemistry called isomers. These isomers are molecules that have the same chemical formula, that have the same elements in the same numbers, but they're hooked together differently. So if you have the same chemical formula, but your elements are hooked together, your atoms are hooked together differently, then you have something called an isomer. And because they're hooked together differently, they will have different shapes, and that will lead to different functions. So even though they have the exact same ingredients, they aren't the exact same thing. Here are 3 monosaccharides. Glucose, which you've all heard of, you might not remember, but you've definitely all heard of it. And then galactose, which most of you probably heard of, but I would think most of you would have forgotten. And fructose, which maybe is half and half. Glucose is the most important simple sugar. Glucose is like key for energy production in all cells. Galactose is a monosaccharide that's found in milk and dairy products. And fructose is found in fruit. And these molecules are found in other things too, but they're really abundant. Like galactose makes up a big part of milk, and fructose makes a big part of fruit. So we can kind of think about difference between these things based on where they're really abundantly found in nature. And all 3 of these have the same chemical formula, C6H12O6. They have 6 carbons, 12 hydrogens, 6 oxygens, but they behave really differently. If you are diabetic and you're experiencing low blood sugar, you need glucose. Could your body use galactose or fructose? Yes, but what it really needs is glucose. So if you give somebody who's diabetic galactose or fructose, their body has to do some work to turn it into glucose before it can really be used. That's why if you go to the store and get pills to give somebody who's diabetic, if they have low blood sugar, they are glucose tablets. It's not just like sugar, because glucose is the most important energy sugar. There are some other considerations with carbohydrates in terms of the vocabulary. So I said that if you take these are monosaccharides, these are simple sugars here. You start hooking them together, you can make a polymer. There's a name for this specific reaction when you polymerize monosaccharides to make a polysaccharide. That is a reaction called dehydration synthesis. Did you study that in chemistry? Does this ring a bell? Yeah, okay. So another term for that is condensation. And basically what happens is that if you take certain things and hook them together, then as they get hooked together, they shed atoms that then reform as water. And that's what happens when you make a polysaccharide. As the enzymes are joined together, water is produced as a byproduct. So you can see what's going on. Oh, no, stop. You can see what's going on here, where we have a hydroxyl group here and a hydroxyl group here on these two. These are probably glucose. I don't actually know what they are, but they're probably glucose. And when this dehydration synthesis occurs, the hydroxyl group from here goes bye-bye. The hydrogen from this hydroxyl group goes bye-bye. The 2 monomers are hooked together with that remaining oxygen in between them, and the hydrogen from one and the hydroxyl from the other combine together to make one molecule of water. And we call the specific connection between 2 monosaccharides, a glycosidic link. So glyc is like sugar, or it's a prefix meaning sugar or sweet, and so this is like a sugar link, basically.
Wait, for the enzymatically connected, like what enzyme is needed?
So I don't know the name of the enzyme off the top of my head, and you won't need to know it either. It's the important thing here is it won't just happen. Like it takes, you need an enzyme to lower the activation energy, or it won't happen on its own. So if you were to take like glucose and fructose and just mix them up together, they wouldn't necessarily combine. But if you have the right enzyme, then they'll be chemically combined to make sucrose. Yeah.
Wait, is that how your body makes like fats with extra sugar?
Yeah, well, so it's a little bit more complicated because in order to turn excess sugar into fat, there's a bunch more chemical reactions that have to take place. But yes, the sugar, the carbon in the sugar ends up being modified and chemically reacted to form lipids, which then are longer term energy storage. Here's an example of a disaccharide. We'll see a couple of these. This is one you're all familiar with, sucrose. This is table sugar. So table sugar is just like if you get sugar at the store, that's what you're getting in general. And it has, it's a disaccharide. There's a bunch of important disaccharides, which means it's a two sugar molecule. It's got two monosaccharides hooked together. And the two monosaccharides are glucose and fructose. So glucose is that really super important one. Fructose is a sugar that is really abundant in fruit. And so glucose plus fructose together, this disaccharide, this is what we get when we get sugar packets at the store, or sugar cubes or something like that. Another really important disaccharide that you're going to need to be familiar with is lactose. And this is, again, something you've looked at before almost certainly. Lactose is one of the more abundant sugars in milk. And it's a disaccharide, just like sucrose is, only this is made from glucose, which is the same thing that's in sucrose, but that other weird one that called galactose. And I think this is funny because like lactose is a really common thing, but galactose just seems like such a crazy, there's something about this that seems like a crazy name. And I think it's because it just seems like something out of Star Wars. Like I imagine this is the name of like a ship in Star Wars. We'll take the galactose, but it's just a sugar. And it's really similar. Like if you look at it, like I could, if you took this diagram and threw it down in front of me and said, what sugar is this? I would never say it's galactose. I would say, well, it's a 6-carbon sugar, it's probably glucose. That would be my guess. I'd be wrong. Look at how similar it is to glucose. Like it's this right here. Like this hydroxyl group and hydrogen are flipped. I think that's the only difference. Like it's a different molecule because of that configuration, that spatial configuration of those two little pieces. All right, and then if we can put carbohydrates, monosaccharides together through dehydration synthesis, then it stands to reason that we would be able to take them apart as well. When we take carbohydrates apart, it's a reaction called hydrolysis. Stop doing that. It's a reaction called hydrolysis, and hydrolysis basically is when you break something apart and a water molecule gets used in the process. It's the opposite of dehydration synthesis. So hydrolysis is when you add something or add water to something, and then that is facilitates the breakage. It's not a physical thing. It's not like you spray this down with water and then it just breaks apart, or you just like put it in water and it breaks apart. It's a chemical reaction involving water breaking apart and other things breaking apart.
Does this only happen to disaccharides?
No, there's hydrolysis can happen to lots of stuff. Hydrolysis is a general thing, but it is what happens to disaccharides and polysaccharides when they get broken down. Yeah.
So if someone like dissolves table sugar in some water, it's still like the long chain.
Yeah, that's not hydrolysis. Yeah, if you then dry that out, you end up with sucrose. Yeah, it's that you in order to hydrolyze, it's a chemical reaction. It would take an enzyme to do this, and so basically the water gets broken apart and attached back where it started, where you get a hydroxyl group here and the hydrogen goes here, and you end up with the same two things that you would have started with to produce the molecule. And this is something where the steps involved in this are like we're not going to need to know any of the steps. This is as much as we need to know. But there would be dozens and dozens. Like if you were a chemist studying this reaction, you'd be looking at way more than just this. Okay, there's a ton going on there. All right, and then the last thing I want to talk about is the physical configuration of monosaccharides and what that means for polysaccharides. So when you look at monosaccharides when they're dry, they exist as linear molecules. So they have two ends, right? And by convention, so all these monosaccharides kind of look the same. And so by convention, we draw them like this, and then we even number the carbons so that they match up like this. So you'll see stuff like this. There's 5 and 3 and 4 carbon sugars, but they'll all kind of have the same general layout when we draw their linear structure. But if you take those and you put them in water, then they bend and then connect in such a way as to form a ring. And so, and the ring is called a cyclical monosaccharide. Nobody uses that term. You should probably know it though. But usually we just call it a ring. Is it in the ring form or linear form? And there's a little video here that I'm not going to play for you, but it's there if you are interested. And because the condensation reactions, those dehydration synthesis reaction occur in water solutions, they're always occurring to things that are in ring forms. So when you look at polysaccharides, they are rings that are connected to other rings. And then even if you dry them out, even if you remove them from water, they don't return to their linear form because now they're kind of locked into place with these other rings. So this is dry monosaccharide. This is monosaccharide in solution. And if you look at polypeptides, it's always the ring form because you don't get polypeptides by combining dry monosaccharides. Okay. So that's a lot, and it's not all. There's a bunch more too. Let me tell you what your job is going to be, both over the next 15 minutes or so of class and outside of class as well. I have 2 things for you to do. What I want you to do during class, and you're not going to get all the way through this, and that's fine. But there is a assignment on Canvas called Building Biological Molecules, Carbohydrates. And if you click on that, you'll see it's like a scan. It's a PDF that's scanned in. And it is asking you to cut some things out and like tape them or glue them together. And you don't need to do that. I have the sheets. So these are the things that they would be asking you to cut out and glue together, but you don't need to do it. You can just draw them. And you don't need to draw all the individual atoms. You can just like draw shorthand versions of these because the point is to see how they hook together and how the different shaped ones hook together because they hook together differently because of their shape. But the whole cutting and pasting that's not really going to give you much for your time and energy. And this is something you can just document this stuff in your notes. So you can work with a partner. And you'll have your green and your yellow sheet in front of you, and you'll figure out which one's the beta and which one's the alpha, and then you will just like answer the questions and figure out like what's going on. And then if it says cut them out and lay them side by side. You don't have to do that, just look at them. And then if it says, tape them together, or glue them together, or whatever, paste the two molecules to your page, you don't have to do that. Just like put the sheets next to each other and just kind of look at them. You'll be able to see this stuff without cutting and pasting. You probably won't have time to get all of this done, and that's okay. Think about your goal here, which is to learn as much biology as you can. What I would do, the way I would approach this, is I would look at the things, and if it's obvious, I wouldn't even do it. Like, what's the name of this covalent bond? I don't even need to read the rest of the question. I know. This is asking about the glycosidic link. So I'm not even going to bother writing that down. There's no point. This linkage is defined as an alpha 1,4 linkage. That's a little bit trickier. That doesn't make any sense. Why is it called an alpha 1,4 linkage? That would be something to talk about with your partner. Why is this a 1,4 linkage? Why is it an alpha 1,4 linkage? Is there a different kind? Is there a beta 1,4 linkage? Like, here's one that's a alpha 1,6. Like, why do we have different names for these different bonds? That's what you should focus on. The stuff that you don't already know. The stuff that doesn't either leap out to you as being obvious, or the things that you didn't already know before you walked in the door. So I will I'm gonna just like let you grab one of these per like pair or so. I need them back, so don't write on them. Definitely don't cut them out. The main sheet is on Canvas. I'll leave it up here too, so you can kind of look at it. But we'll just spend like 10 minutes taking a look at this. I would, like I said, focus on the things that you don't know, and then after we're done with that, then we'll move on, and I'll tell you what the last little bit is, which is a homework assignment. Okay. Does anybody have any questions before you get started?